Can I start my speech, sir?
Yes, yes. The first speaker of today is Professor Shamapada Mandal from University of Gaurbanga, India, and he is going to give a presentation on perspectives to the COVID-19 immunopathology and vaccine status. And I request Professor Mandal to start his presentation now.
Thank you very much. This is Global Webinar on Immunology 2021, and I am speaking Shamapada Mandal from Gaurbanga University, Department of Zoology, India. My talk title is Perspectives to the COVID-19 Immunopathology and Vaccine Status. So what is the scope and background? Background is COVID-19 pandemic. We know this has been started in the year 2019 in the month of December in China first, and thereafter in March 11 World Health Organization declared the disease COVID-19 as a pandemic. That means this pandemic is still going on, and there are several kinds of devastation due to the COVID, and still we are not working normally. We are working from home, and there are very difficulties with getting to the normalcy, and the disease giving different types of conditions, signs and symptoms, and different types of difficulties. And one of the most important is cytokine storm that is related to immunopathology of SARS-CoV-2 infection. SARS-CoV-2 means this is causing COVID-19 pandemic. And COVID-19 vaccine, we know that there is no such vaccine that can prevent the COVID-19 pandemic, as well as no specific drug to treat the disease COVID-19. So at first you have to know the strength of the pandemic. This is long pandemic because 1 year, more than 1 year has been passed, and this is also characterized the long COVID. That means long haulers are present. Some patients are there, after even they got negative test to the PCR, they are still bearing some characteristics, some signs and symptoms and difficulties, clinical features. So these are long haulers. So COVID-19 pandemic is causing even now, the devastating situation. And in the world scenario, this is the scale of the cases. Today we are seeing that the total cases is 126.7 million, and death is 2.8 nearly million. And the vaccination has been started, and this is the global scenario, and this is the death scenario. The total death is 2.7 according to 24th scenario of the COVID-19. And this is top 10 countries. This is global, I have already said. And first is United States, then Brazil, India, Russia, France, United Kingdom, Italy, Spain, Turkey, and Germany. These rank is according to the number of cases in that countries. And this is the new cases reported. We are seeing that in United States 60,000, in Brazil 85,000, in India 68,000. These are havoc and causing havoc and damage due to new cases. And the death is like this that I have already told. The global situation, this is the United States, 0.54 million, Brazil 0.31 million, India 0.161 million, top 3 countries. And this is newly death. India 291 new death number. So this is low in number compared to the other countries, except Turkey or Spain. There is no death case, or in Germany or in United Kingdom, which is 158. And their transmission pattern, community transmission majorly seen in United States and Brazil, and in India it is cluster of cases. Community transmission is also available in France and United Kingdom, and likewise these are community and cluster cases in different countries, in top 10 countries. Here are 6 regions of WHO, World Health Organization. First is Americas, then Europe, Southeast Asia, here India is located, Eastern Mediterranean, and Africa, Western Pacific. This is the number of cases of COVID-19. Top is Americas, 55 million, then Europe 45 million, Southeast Asia 14.69 million, Eastern Mediterranean 7.43 million, Africa 3.06 million, Western Pacific 1.873 million. And this is the death cases in different WHO regions. First is American, the Europe, and then Southeast Asia, Eastern Mediterranean, Africa, and Western Pacific. And this is Gavi region. So we know there are 4 regions and 73 countries have been signed to Gavi for vaccine distribution in developing countries, actually the poor countries in the world. And these are Anglophone Africa, Asia Pacific, EMRO, Eastern Mediterranean, Europe region of WHO, and Pan American Health Organization, and Francophone Africa. The number of cases are presented in graph, both graph, according to March 29 of this year. Asia Pacific remains in the top. This is Asia Pacific, this is Asia Pacific, and accordingly the death cases are also higher in Asia Pacific comparative to the other 3 Gavi regions. And here are the top 12 regions. First India, then Indonesia, Pakistan, Bangladesh, Georgia, according to the number of cases. So we know that 73 countries have been signed for Gavi for vaccination in poor countries, that developing countries, for children specifically. And here WHO COVID cases in different regions, top 6 regions, top 3 countries, United States first, then Brazil, India. This is the pattern of cases. So we are seeing that India is now increasing in number of cases. Already this has been told that the 29th, that yesterday the number was 68,000, more than 68,000. But Russia, this is decreased. France, this is somewhat increased. United Kingdoms decreased. Brazil somewhat increased. And this is the death situation in India. Took the 4th position. This is the number, but cases is 261. This is comparatively lower compared to Brazil, Mexico, United States, Italy, and in United Kingdom the death number is also low, and this is lower than India also. And we know that even though 1 year has been passed, but there is no specific drug for the treatment of COVID-19 pandemic that I have already told. And there are some drugs for the treatment of parasitic infection, other viral infection, bacterial infection, fungal infection, and cancer treatment. So these already available drugs are repurposed for the treatment of COVID-19, and there are different druggable targets virally and related to host also. Related to host is ACE2, angiotensin converting enzyme 2, and TMP-RSS2. These are inhibited by nafamostat, camostat, and chloroquine. Though there is some adverse report, but chloroquine is can be applied in the treatment of COVID-19. Endocytosis is inhibited by imatinib. That means this inhibit the entry of the virus by endocytosis, inhibited by chloroquine, hydroxychloroquine. And there are PL polyprotein 1A and polyprotein 1AB. This is hydrolyzed by 2 proteins, 3CL-Pro and PL-Pro. These are very important enzyme for the life cycle of the virus SARS-CoV-2. And these 2 enzymes are inhibited by lopinavir, ritonavir first, and then for PL-Pro, disulfiram has been applied for PL-Pro inhibition. And the enzyme, viral enzyme, RNA-dependent RNA polymerase. This is a very important enzyme in replication transcription complex, and this is inhibited by remdesivir, favipiravir. And cyclosporin A, this is antifungal drug. This also inhibits somewhat in the process of translation and transcription, and so that the viral structural proteins ACAN are not assembled, and viral particles are not formed. And if formed, these are released and then cause cytokine storm, and these are due to release of IL-6R, IL-6, and this is inhibited by the receptor. IL-6 receptor is inhibited by sarilumab and tocilizumab. And why? How? And this is very devastating, because we know these are derived from the genetically from animal, that bat to human. through maybe pangolin, and then now transmission from man to man for the last 1 year or more than 1 year is occurring, and aerosol or droplets are the cause. That means due to sneezing, coughing, etc. Droplets are released, aerosols are released that are related with the viral particles and then cause transmission through fomites, contact, and etc. And these are inhibited by the use of face masks, since there are no drugs or except remdesivir, which is approved by FDA, and no vaccine is has been developed for the prevention of the transmission of COVID-19. The causative agent is SARS-CoV-2. So that this is devastating, causing different types of symptoms such as firstly fever, gastrointestinal issues, myalgia or arthralgia, and dry cough. This is the first symptoms, shortness of breath and body ache, and other symptoms include headache, fatigue, loss of smell, sore throat, chills, and loss of taste. These are the main features, main clinical features, and still we know that the cytokine storm is the cause that cause different organ damage, and the immunopathology of the SARS-CoV-2 infection is like this. The immunopathology of COVID-19, lymphopenia, all kinds of lymphocytes are decreased in number, and these are exhausted due to release of high number of interleukin-1 tumor necrosis factor alpha, interferon alpha, and these are also dysfunction. T cell exhaustion occur and natural killer cell exhaustion occur. And here we are seeing that number of granulocytes, neutrophil, eosinophil, and basophil. Neutrophil is increased, granulocytes, eosinophil, and basophil are decreased, whether non-cyte monocytes increased in number. That's that large number of cytokines, mainly
Yes, it's,
it is very visible that probably started that I have dropped potential mechanism of SARS-CoV-2 induced immunopathology. Here, impairment of T cells, T cell depletion occurs, lymphocytes, the lymphopenia caused. I have already told T cell impaired due to dysfunction and highly function. And here, due to this situation, due to lymphopenia, other kinds of viral infection or fungal infection or secondary bacterial infection occurs due to lymphopenia. And so microbial infection increased. And in this situation here, we are seeing a cytokine storm, mainly due to IL-6, IL-10, IL-1 beta, and here is also IL-17 is also increase in cytokine storm is caused, and antibody dependent enhancement is caused because here we are seeing that cells are activated and infected due to SARS-CoV-2, and the number of cells are increased, so the disease condition is become poor and it progress. Here immunopathology of COVID-19, already lymphopenia I have already told, and this is activation of T cell, this is also told, and lymphocyte dysfunctions occur. And here I have seen that interleukin beta, IL-6 and IL-10 and number of immunoglobulin, total antibodies are increased due to dysfunction or, of lymphocytes, and monocytes are decreased. So I have already told, I think, and here the clinical implication, the main implication due to cytokine storm is lung, that wherein causes ARDS, respiratory failure, and for liver, ALT, AST, and gamma GT is increased, causing liver damage. Kidney damage is also occur due to increase of urea and creatine kinase and lactate dehydration increase that cause the effect of heart. So not only the lung, liver, kidney, and heart damage are also occur due to cytokine storm. And we know that probiotics, mainly the lactic acid bacteria, bifidobacterium, are used because these they have function against dysbiosis, and it maintains immune homeostasis, because we know that some probiotics are translocated in the intestinal wall, and here this dysbiosis occurs due to the SARS-CoV-2 infection. But if we apply from outside probiotics, then SARS-CoV-2 infection is decreased, and lung damage or other organ damage is decreased also. And here actually brain gut probiotic brain gut axis is maintained, because we know that probiotics required for their growth and function in prebiotics. Prebiotics are some functions that are selectively used by probiotics, and here psychobiotics, that means the probiotics and prebiotics, when related to neural disorder, again causing central, that means when it maintains blood-brain barrier due to the disruption of COVID-19 infection, SARS-CoV-2 infection, that vaccine wherein vaccination has been started globally, and America's top, and India's situation is like this. India already already administered 54.66 million doses, and the system has been started in 16 January. This is number of vaccines. We know three vaccines in India already approved, two by Hello, is it okay?
Yeah, please continue.
So we are seeing the different types of vaccines are available. That may be weekend virus and inactivated virus. These are whole vaccines, and component vaccines are also available, such as replicating viral vaccine. These can replicate inside the patient, and non-replicating the adenovirus, and in case of replicating measles virus are utilized. And DNA and RNA vaccines are available for nucleic acid vaccine. These are nucleic vaccine, and some kinds of proteins, subunits, as well as viral-like proteins. That means this is not a virus, SARS-CoV-2. This is created with protein showing the structure of SARS-CoV-2, and when administered, it can cause immunogenicity. So the different kinds of vaccines are presented herein that I have already told. This is in summary. And globally, there are 12 vaccines are available, RNA, and these are approved. Viral vectors, protein and inactivated virus. Moderna, these are treated by Moderna, and BioNTech, AstraZeneca, Janssen & Janssen, CanSino, Gamino, and Medicago, Novavax, Bharat Biochem, Sinopharm, Sinopharm, and Sinovac. And highest activity or efficacy has been achieved in BioNTech 95% and Moderna 95%. Ranking, according to ranking, RNA ranks first in terms of safety, efficacy, logistics, COGS, and total score is 82. Whereas viral vector score is 74, protein vector 80%, and inactivated virus vector 1%. And you are seeing that 184 vaccines are globally available under clinical trial, and 29 vaccines for phase 2, 28 vaccine for phase 2, and 21 vaccine for phase 3, and 13 are approved, and 5 vaccines are not continued. And we are seeing that different phases of vaccines are available in the
world, and in case of WHO, 13 vaccines are continued. These are RBD dimer, Covaxin, AD5, that means adenovirus 5 nCoV, Epivac Corona, Sputnik V, AD26 CoV2S, mRNA 1273, AZ1, that means AstraZeneca, Oxford AstraZeneca, AZD122, BNT162B2, Covishield, BBIB, CoV inactivated varicelles, and World Health Organization approved 4 vaccines, of which Covishield, that is created in India, are now continued. And in India, 10 vaccine, which is Oxford AstraZeneca, Sputnik V, Covishield, BBV150, Covaxin, and Biological E Limited created 3 in India, Bekov 2A, Bekov 20B, Bekov 2C, and Bekov 2D, Jairus Cadila. That means 10 vaccines are available under clinical trial in India, and of which 3 are approved and 2 approved by World Health Organization, Covishield and Oxford AstraZeneca, and other is Covaxin created by Bharat Biotech. And now these are available for treatment, for prevention. And Covishield, this is CHAD-OX1 nCoV2. These dosage is 5.10. 2 doses are applied in at least 4 weeks gaps. And special precaution storage is very nice in normal fridge temperature plus 2 degrees Celsius and 8 degrees Celsius. And Covishield, this is also applicable in case of patients carrying or causing infected with the variant B1351. Covishield is important. And Covaxin, this it has been reported that 81% efficacy rate has been achieved due to Covaxin, which is available in India and distributed. And now at last we are seeing that there is no still proper vaccine for the prevention of transmission of COVID-19, and the situation is very devastating till now. So with a fast moving pandemic like COVID-19, which is ongoing for last more than 1 year, there is no drugs or vaccine, so we are not safe. And with a fast moving pandemic, no one safe is unless everyone safe. So lastly, I acknowledge Global Webinar on Immunology 2021 Organizing Committee, Global Scientific Guild, for recognition of my research and supporting me for this presentation. Thank you. Thank you. Stay safe, stay healthy. Thank you, sir.
Thank you. Thank you, Professor Mandal. Thank you for your excellent presentation.
Yes, thank you.
Yes. I have a question for you.
Yes.
Can you please tell me how efficient is Bharat Biotech's Covaxin?
Yes, I have, as report, Covaxin is efficient, and it has been reported, just I have told that 81%, but some side effects are available. But when second dose are applied after 4 weeks or more than a gap of 4 weeks, when gap is large, that means more than 4 weeks, 12 weeks, it is fine, and no side effects. That means side effects are milder. So it is working, but it may be varied person to person because of different types of immunogenicity. That means some persons may not be tolerated with that, but these situations are low in number, since 81% efficacy has been achieved by this vaccine, as well as Oxford AstraZeneca. So these vaccines are working, but I don't know. I have not seen the report how many days it will persist in the human body after administration.
Which one will you prefer between Covaxin and Covishield?
There is no preparation because there is no choice. These are producing in a daily rate, and not available to others. Only I have told that 50 less than 60,000 million doses have been distributed. So we cannot choose in such and such way. Just we have to wait to take. Both are working: AstraZeneca, Oxford AstraZeneca, and Covaxin.
Okay, thank you so much. Yes, Dr. D.N. Rao. Dr. Marco.
I was amazed that several kinds of vaccines are on the way.
Yes.
Everybody in Europe thinks that the amount of possible vaccines is far more restricted. So hopefully we can get some in clinical use as soon as possible, right?
Yes. So if you share your question.
Can you please type your question, Dr. Marco?
I was wondering how close your several vaccines, despite the known, how close they are to clinical use, to translation.
I'm not getting your question, so please type this.
Can you please
differently, because the system or the doses of vaccines are different, and the procedure of development is different, and storage process, distribution process, and country-wise this is different. So these are not working in a same pattern. Okay, and not all are active as we have reported, as we have seen in report.
Okay.
Still, we have to wait to get the effective virus, because we know that the variants are developing, emerging, SARS-CoV-2. So there is an effect on the vaccine development due to variants in different countries, in India also. But reports are available with Oxford AstraZeneca. These are also effective against new variant, which are found in India.
Okay.
Would Dr. Rao like to share some input in this issue?
Yes, it will be fine for me also. We will gather some knowledge from him also. And yes.
The second wave has started in India. How different the second wave will be from the first wave? How efficiently can second wave can be control it?
As I have seen the reports, this is not due to variant. This is a new wave due to the same virus actually in different places. Variants are very few in number in different parts of the country. So it can be built because we have gathered some knowledge to control, because we are using mask, we are maintaining distance, we are getting some vaccines which are approved in India. One is produced in India that are also approved by World Health Organization, and India approved 3 vaccines, and the companies are available for the production of a large number of vaccine doses. So it can be controlled, but we have to wait and see how much time will take, and some experiments will also needed for simulation, as we have published earlier when it was in first wave. We have published some papers. So we have to see the pattern of development of increment of number of death also, not only the cases.
How different this will be from the first wave?
Different is that this is still increasing, still in presence of a little bit of availability of vaccine. So if it will be fast if we do not ignore the situation, if we maintain distance, if we wear mask. Otherwise, it will be more than devastating that was caused in the first wave.
And how much prepared the medical experts are from the first wave for the second wave? How much they are prepared?
Yes, yes, they are prepared because PPE are available, and and there were no training for COVID-19 in World Health Organization and other such as ICMR and India, who have given different types of training program. So and which is most related to this pandemic. So I think we are prepared, and personally the publics are also knowing the situation. They know better than before, so they personally can control the situation also, not only the frontline clinicians or the doctors or nurses.
Thank you so much for answering the questions, Professor Mandal.
I don't see the.
Just a minute. Yes.
This is the aroma of immunology. Actually, I can explain to you. For inducing an antibody response to any protein, the protein should be long enough, and you should carry multiple antigenic determinants. That's what the previous speaker told. It must have enough epitopes to contribute a population of antibody. That's very important. If you see that third one, you can see many epitopes are there. Mostly the proteins have large number of epitopes. That is the reason protein is considered to be the best immunogen when compared to any other thing. If you take lipids or polysaccharides, none are much immunogenic, but proteins are considered to be highly immunogenic. This is because it carries a large number of the epitopic configuration. Next slide, please. This is actually the previous talk essence actually. For inducing a protective immune response to any vaccine, the genetics of an individual plays a major role. This is why where we have to kept in mind throughout our as long as the immunology survives, you have to remember this statement. I am giving a simple protein here. It is a cartoon which has this is a protein. When a protein goes into the body, it undergoes the proteolytic cleavage and gives rise to a large number of epitopes or antigenic determinants. This is normally takes place in the antigen presenting cells, and this epitope is taken by the individual MHC molecule, individual MHC molecule or HLA molecule, and finally presents to the B cell or T cell to induce either the cell-mediated immunity or the humoral response. This is the crux of immunology. Why many vaccines are failed, why many vaccines shows a variation in individual is because of this individual genetics actually. How the process, how a protein is degenerated into different epitopes, how it has been picked up by individual human leukocytic antigen, HLA antigen, how it is present to the B cell and T cell. That is actually the crux why I am producing a very high antibody levels, whereas somebody is not producing antibody response to the same vaccine. This is actually the crux of immunology actually. Next slide, please. This is how it is. If it is a protein, the antibody reads a certain region of the amino acids. That's how it induces the what you call the antibody protection. Next slide, please. And if you come to the subsequent slides, I used the enzyme linked immunoassay for detection of antibodies in a different set of people using the what you call the Chikungunya virus epitopes. And today this has been amplified by using chemiluminescence method. If you look at most of the diagnostic approaches, chemiluminescence method is the one which is being routinely used because it is more sensitive and it is more sensitive. But enzyme immunoassay has its own advantage because this can be operated in a simple lab, and later this can be translated using a chemiluminescence technique. Next slide, please. I don't want to tell much into this disease. Actually, chikungunya is one of the disease where people are once they infected with the disease by the mosquito bite, you end up with fever, headache, and nausea, and mostly the joints are affected. Actually, joints, what you can see is the large amount of body pains, joint pains, etc., etc. One can see in this disease. This comes under the category of the what you call Togoviridia is the genus of the disease actually. Next slide, please. And they normally it is a self-healing disease. You don't have any medicines actually for this. It is a self-healing disease. Normally incubation period is 4 to 7 days. Normally by 7th day or 10th day, the symptoms of the disease stabilizes. And this more or less, there are 3 similar viruses are there that have been taught earlier, dengue, Zika, all this comes under the same category of whole thing actually. Next slide, please. Next. This is the global scenario where the disease is evenly distributed throughout the world. Next slide, please. And this I want to skip actually. Next slide, please. This is how it is actually. It is a mosquito-borne disease. And normally, the daytime mosquito is the one responsible for the bite of this mosquito gives rise to the disease symptoms. It has to carry. What it has to do is it has to carry the virus from an infected person, and it is transmitted to a healthy person. Next slide, please. And this is actually, many people believe that this might have came from the monkeys also, but we don't have much details on this. But it is a disease, a global disease where it is transmitted from individual to individual, not from the monkeys or sylvatic cycle. Next slide, please. This is the structure of the virus actually. If you look at the structure of the virus, it encodes 4 non-structural proteins, the NSP1, NSP, P3, P4, like that. They're all non-structural protein. They're no way involved in as far as the protection is concerned, they're no way involved. The major involvement of the protective antibodies and target of immunity are involved with the E1, E2, and E3 proteins. These are the 3 important proteins, very important as far as the protection is concerned, as far as inducing the antibody in the patient is concerned. That is the reason today most of the people are working using these are called structural proteins. People are using these structural protein, either E1, E2, E3, a combination of all the 3 proteins for inducing the protective immunity, as well for diagnostics also. Today, whatever diagnosis you are seeing in chikungunya, this is based on E2 protein only. They immobilize the E2 protein and they look for anti-E2 protein antibodies, and finally they say the diagnosis, etc. So the structural proteins plays a major role both for diagnosis as well as vaccine design. Next slide, please. I'm not going much into the details. Just you can skip this slide. Next slide, please. Next slide, please. This is something about the length of the viruses. It is a single stranded RNA virus, and there it gives rise to 2 types of proteins, non-structural and structural proteins. And structural proteins are the one which are involved in both for diagnosis as for the vaccine design. Next slide, please. This is the immunology of the virus actually. The moment if it is infected with a healthy person, there is an increase in the viral load, and this is followed by type 1 interferons. When the T cell produces type 1 interferons, you can see the suppression of the virus actually. That's what you can see. And if you go for more days, like after 5 days or 6 days, you can see the production of antibodies. So initially, there is a strong T cell response which restricts the growth of the virus. Later, the same body induces the B cell response in the form of antibodies that protects the person. That's the reason it is called a self-healing disease. You must require a very strong T cell response as well as a B cell response. Next slide. So since there is no vaccine currently available what you thought actually that time, almost all this study was done almost all 8 years back. And we thought actually, why can't you go a simplistic method? Make make this protein as a target and look for the antiprotein antibodies in the serum of the infected people. Of course, there are molecular techniques are also available currently. This is more expensive. RT-PCR is there, but is expensive. So serology we preferred. This is because we can look at the antibody profile in the patients using a specific antigen. Next slide, please. Next slide, please. Next slide, please. What we did actually initially is we don't know really what are the epitopes that induces antibodies in a given individual when a vaccine is given to you. So since we know something about the structure of the virus, we know E1 protein, you know E2 protein, you know E3 protein. All three proteins are very important as far as the immunity is concerned. And especially we came to know that E2 protein has a receptors on the antigen presenting cells. Once it binds on the receptor on the antigen presenting cell, it undergoes internalization by endocytosis and starts replicating and giving rise to neuron. Hence, we focused our attention mostly the E2 protein to see whether we can detect anti-E2 antibodies in the infected individuals. Next slide, please. And the same thing, same slide only. Next slide, please. What we did actually is there is a computer-aided programs are available to scan the protein. If you scan the entire protein, E2 protein, and you can see what are the sites for immunogenicity. But because the previous speaker already used that word, it depends. The antibody profile mostly depends on the immunogenicity of that protein. So what we did actually, we want to see what are the areas which are found to be highly immunogenic, so that we can synthesize those immunogenic epitopes in the laboratory, so that we can use it for the serodiagnosis. So this is the program we used it for a DNA star program to map the vulnerable antigenic sites on the protein. Next slide, please. Next slide. Next slide also. This is E2 protein. This is E3 protein. So we know comparatively what are the immunogenic sites or antigenic sites on a protein where you can pick up and use it as an antigen for diagnosis. Next slide, please. This is the Pymal study. It is a computer-aided Pymal study. What we did, there is a entire protein is there, and we want to see actually where are this. We have predicted almost all 15 to 20 epitopes on E2 protein, and we want to see whether this E2 whether we want to see whether these epitopes whether they are present inside the protein or outside the protein. Mostly, if a protein is outside and if the epitope is outside, then immune system nicely recognizes. If it is internal, it is very difficult, hardly is very difficult to recognize the internal structure of the proteins. Hence, we studied a Pymal study where you can see where is this epitopes are lying. Mostly the epitopes are present on the surface of the protein. Hence, we thought maybe it is a wonderful idea to go for the diagnosis of the infected persons. Next slide, please. Next slide, please. Next slide, please. Next slide. So what we did actually is we initially standardized the ELISA by quoting different concentration of the E1, E2, E3 peptides actually or epitopes, and we did a classical immunoassay. We have taken almost 125 patient sera. Those who are coming to the hospital, and these are known positive cases. These are known positive cases. These patients sera has been taken, and at different dilutions we used for detecting the antigen antibody complex using a ELISA program. Next slide, please. This is we measured both IgM and IgG antibody. IgM is considered to be a beginning antibody. Whenever you get an infection, whenever you take a vaccine, initially you get IgM type of antibodies, and after some time, after taking a booster, second immunization, it shifts into IgG response. IgG antibodies is considered to be highly protective. That is the reason we look for both IgM and IgG antibodies in these patients. Next slide, please. Next slide, please. Next slide, please. This is what we did. We have taken E1 and E3 peptides, epitopes, what we synthesized in the laboratory, and these have been coated on the ELISA plates, and we have taken E1 protein. We have made a recombinant E1 protein also in the laboratory, and we have made a viral lysate also. Viral lysate also we have taken, and all these two are taken as a positive standard, and E1 and E3 peptides they are taken, and we looked for a antibody profile. Unfortunately, none of the patients here do recognize either E1 or E3 protein peptides, whereas the proteins and viral lysate recognizes protein, and to some extent the total protein and the viral lysate recognizes, whereas if you could see each epitope, they don't recognize the protein, and each epitope is almost all 20 to 25 amino acid length. Next slide, please. Next slide, please. Next slide, please. When you quoted the different E2 protein, E2 peptide on the ELISA plate and looked for antibody, you can see very nicely antibody profile. It is a IgM antibodies you can see, and this is compared with the total protein as well as the lysate also. And this clearly indicates us when you taken a patient sera and look for antibody profile, mostly the antibodies are directed against E2 protein only, not for E1 or E3 protein. This clearly indicated as this can be exploited for diagnosis. Next slide, please. And this is IgG profile. If you look at for both IgM and IgG, you can see both the type of pattern of immunoglobulins you can see. Mostly some people are early phase people, some people are late phase people. That is the reason you can see both IgM as well as IgG type of antibodies in these patients. Next slide, please. Next slide, please. We look for IgG type of antibodies, very important in immunology. Instead of looking for IgM and IgG, there is a isotyping is there. Within IgG, there is 4 isotypes are there. Out of these 4 isotypes, IgG3 is the best antibody. As far as the neutralization of the virus is concerned, killing of the virus is concerned, IgG3 antibodies are the best. So we looked for IgG3 type of antibodies, and most of the individual do carry the IgG3 type of isotypic antibodies in this patient who felt that these people have a protective antibody as far as the clearance of the virus is concerned. Next slide, please. And when all the epitopes were taken 123 sera, and each epitope was reacted with the patient sera and looked for sensitivity and specificity, etc. When you looked for different epitopes against different epitopes, we looked for antibody profile, and slightly we got disappointed actually. You could see actually the out of 123 sera we collected, some showing 115 positive, some showing 100 positive, some showing 93 positive, and there are some missed cases are also we could able to see when you're using epitopes, etc. Slightly we were disappointed, but if you see the specificity and sensitivity, they're really very good. They showed a very good high specificity and sensitivity. Only disappointment came for us is not able to pick up each epitope is not able to pick up individual sera when you look for so many patients sera. Next slide, please. Next slide, please. Then this is the one of the, when you look for IgM, IgG, IgG3, all type of antibody, somehow this is a big disappointment. And the total protein, E1 and E2, we got it from the virology department of DRDO Gwalior. And next slide, please. And what we did actually, those epitopes showing above 90% reactivity with the patient sera, they're all been clubbed actually. They made as a recombinant protein. So 4 epitopes has made a recombinant protein, and that this we tested actually. Next slide, please. Next slide, please. I will show you the test. If you see the total protein and you see the recombinant protein, those who showed 90% reactivity, 89% reactivity, and 93%, all have been clubbed up and tried for the zero reactivity. And we found that MAP1, there are 4 epitopes which are combined together as a recombinant protein that shows a parallel immunoreactivity as that of E2 protein. This really promised as one of the best immunogen or best diagnostic epitope what can exploit for this one. Later, this epitope this MAP1 was given to some industrial company where they exploited for diagnosis of the chikungunya patients actually. Next slide, please. This is what it is, IgG antibodies. Next slide, please. So if you look at the multiple epitope story, you can see the MAP1 is showing almost 100% sensitivity and specificity, whereas other combinations, they are not showed much this. So we thought MAP1 could be a better antigen for as well as the zero diagnosis is concerned. This has been later translated to a company, and later they made it they made a better refinement of the antigen for various diagnostic purposes. Next slide, please. So this next slide, the last slide. So this clearly indicates that instead of using a single epitope for a diagnosis, if you can join as a multiple epitope, that can satisfy for most of the diagnostic property. You don't know, you cannot miss certain cases also. That is really appear to be a future diagnostic carrier for chikungunya. So we are thankful to the CSR and DRDO for providing the financial assistance to carry out the projects. And this protein is already, this peptide is already with the company already. Next slide, please. Thank you, please. If you have any questions, I can clarify the doubts. If anybody has any questions, I can clarify the doubts.
Dr. Umberto, welcome to the meeting. Please unmute yourself to ask a question.
Yes.
Yes.
You hear me?
Yes. Please continue. Yes, we can.
Yeah. Thanks very much for having me, and I look forward to give my talk in a short while.
Yes. Do you have any questions for Dr. D.N. Rao?
No, I mean, I think the presentation was extremely clear, and I thank you for your presentation. It was very interesting. Thank you.
Thank you very much. Anybody? Swapna, any doubts?
Am I audible? Audible?
Yes, you are audible. Yes.
Clear.
Oh, thank you, Dr. Rao, for your beautiful presentation. I just wanted to know to which receptor of the APC does E2 bind? That's one question. And another question is just a wild thought that whether where the T cells decline, the curve you showed, the antibody raises and the T cell declines. Can you think of some immunotherapeutic measure to stimulate the memory T cells and have some immunotherapeutic effect?
Okay. Coming to the second point, actually, initially when the virus infects a healthy person, what really happens is that you require a very strong CD8 T cell response, CD8, and that gives CD8 T cell response. Instead of having a response, you must to restrict the viral growth. You require a very strong CD8 response is important. That's what really happens. If you measure the CD8 population by flow cytometry, you can see a very nice increase in CD8. As long as you have a strong CD8 response, you have a restriction of the virus actually. They don't multiply actually. Later, what really happens is CD4 cells comes into the play, and this CD4 cells helps the B cell to produce the antibody. That's what really happens. That's what exactly. That is the reason initially you require a strong T cell response in the form of type 1 interferon. Normally teaching the type 1 interferon are the one which is helpful for the restriction of the virus actually. Type 1
can you please clarify that in some cases of fungal infection, some CD4 cell types are cytotoxic. Does the same thing happen in the viral cases?
CD4 T cells are cytotoxic. The
Yes,
cytotoxic
some subtypes.
Actually,
there are some subtypes where CD4 T cells become cytotoxic in case of Cryptococcus neoformans. We have also have experimental evidence of that. But does that happen to the virus type?
This and I don't know actually, but what we observed actually is by flow cytometry, we collected the peripheral and looked for the population, the monocyte population, B cell population, T cell population. Within the T cells, we got for antibody against CD4, CD8, and we gated against CD4, CD8. When we found those people who have restricted the growth of the virus, they have a high number of CD8 population. This clearly indicates that these are the people, they are not the victims of the disease actually. So they require much more faster actually. And asking the first question actually, we did a competitive immunoassay study, competitive immunoassay. So what we did actually is we taken the macrophage saline and incubated with the E2 protein and competed with the different epitopes actually. And we found one sequence which partially displaced the E2 protein. And we consider that this is the epitope which is responsible for binding to the receptor that is present on the APC actually. So competitive immunoassay told us we can map the actual region of the E2 protein where it really binds on the E2.
Okay. But that receptor hasn't been identified as yet.
Yes, correct.
Okay. Thank you so much. Thank you very much.
Thank you very much for patience hearing. Actually.
Thank you. Thank you so much, Dr. Rienro. It was really an excellent presentation.
Thank you very much.
Thank you so much. Yes. Moving on, our next speaker is Dr. Umberto Rossetto from National Cancer Center for Biotechnology, Spain. And I request, and he's going to give a talk on role of GAD445B in hepatocellular carcinoma development. And I request Dr. Umberto to start his presentation now.
Okay, share. You see everything, right? Correct? Okay, 1 second only. I think that should go full screen. So, okay, perfect. So thanks very much for inviting me to the global webinar of immunology. It's a pleasure for me to be here. And I am Umberto Rossetto. I was actually associated with Weill Cornell up until November 2020. But what I want to describe you today, it's actually work that I did in the National Center for Biotechnology in Madrid that actually is going to be published very soon. So I'm very happy to tell you the story. And it's the story about, a gene that was studying in my lab, which is called GAD45 beta. And we wanted to address the role of this gene in hepatocellular carcinoma HCC development. So first of all, just to give you a small background of hepatocellular carcinoma, it represents the third most common cause of cancer-led cell death. Okay, we have 1 million cases per year. The survival rate, if you diagnose it early, it's only 30%. And if you diagnose it late, we go into the single digits here. So it's very important to understand the disease and to find good therapy that can improve survival. So far, we have only one drug approved, which is actually sorafenib, that only add 1 or 2 months to the lifespan of the patient. One thing that is important to realize is that usually hepatocellular carcinoma doesn't really develop alone. You need a chronic source of liver stress for HCC to develop. And for instance, hepatitis B or hepatitis C or even cirrhosis, they create a persistent inflammatory environment that can actually facilitate hepatocellular carcinoma development. So sorry, this slide. So a little bit of detail about the proteins that we're studying in my lab. They are actually the GAD45 family protein. GAD45 stands for grow arrest and DNA damage inducible protein. as the name suggests, these proteins are highly induced following genotoxic stress, and they mediate a wide variety of processes connecting actually with DNA damage repair, like they mediate genomic stability, apoptosis, DNA repair, and influence actually cell death and proliferation. One important thing that they have is that they have a conserved interaction with the MAP kinase cascade. The MAP kinase cascade, it's one of the most important cascade in biology, and it's actually made up of 3 layers. You know, the MAP3K, they phosphorylate the MAP2K, they phosphorylate the MAPK, and phosphorylate many proteins involved in mediating many cellular processes, okay? And what we know is that the GAD45 family protein can interact at various levels with this cascade. I mean, for instance, we know that GAD45 alpha can inhibit p38 activation in T cells, but also in liver, GAD45 beta can inhibit MEK7, and all the GAD45 family members, that are GAD45 A, B, and G, can interact with MAP3 kinases. So they modulate this cascade at various levels. So why did we decide to go for GAD45 beta and hepatocellular carcinoma? What made us think that? Because we check actually in the literature, and there were some statistics that GAD45 beta is downregulated and the promoter is methylated in hepatocellular carcinoma. And also, if you remember what I told you before, sorafenib is the only drug approved for HCC treatment, and GAD45 beta is required for sorafenib-induced apoptosis. So we thought, okay, maybe this gene can actually be a tumor suppressor gene in HCC development. So, but we lacked the evidence. Let's find the evidence. One problem that we had is that, you know, the GAD45 beta knockout mice, they do not spontaneously develop tumor, okay? It's okay, they don't develop, so we need a way to induce. If you actually check the field of HCC, one model that is used to induce cancer is the Atyl nitrosamine model. This is widely used in hepatocellular carcinoma. What happens is that you inject the Atyl nitrosamine in a mouse that is 2 weeks old, you wait for 9 months, and pretty much 100% of the wild type, they develop hepatocellular carcinoma. The way it works is that you inject DAN, you have DNA adduct in some cells, then you have a wave of cell death that kills many, but not all the cells with DNA adducts. Then you have a wave of compensatory proliferation, and as you can see here, there are some mutations here that over the long term, they get selected and they give rise to cancer. This was actually the model that we decided to do. And what we did, we took 2 cohorts, wild type and GAD45 beta knockout mice. We injected with DAN, we waited for 9 months, and this was the first graph that we got that to us was pretty surprising, you know, because 30% of the mice was actually tumor-free. This was contrary to our hypothesis. We got 30% of the mice that are tumor-free, only 5% of the wild type developed tumors. This is a representative picture here that you can see the liver of GAD45 beta knockout mice. It looks completely clean. This is the liver of wild type with DAN. And also in the tumor positive mice, if you notice, 60% of the GAD45 beta knockout mice develop actually less than 10 tumor, compared with 35% of the wild type, and only no GAD45 beta knockout mice develop more than 20 tumors. So there were already less tumor and less tumor per mice. Before hepatocellular carcinoma, you have adenoma, which is a benign lesion that happens in the liver, okay? And all the tumors that we could identify in the GAD45 beta knockout mice, they were adenomas. To our surprise, we couldn't identify any single HCC in the wild type cohorts that we injected. It was pretty surprising. So they say that they are resistant to tumor development. So why was that? We went back to the initial response for DAN because, you know, all these processes required for tumor to be formed. But if you have defect here that the cells cannot die for whatever reason, your gene influences cell death, or your gene influences the proliferation, and both of these processes are blocked, the result is the same. You don't develop tumor. So we checked the early phase, like then induced cell death and then induced compensatory proliferation, because we thought that these processes may be impaired. So one important remark that I have to make is that you inject DAN, you have cell death, and then you have proliferation that leads right to cancer. These processes are intimately connected. You inject DAN, hepatocytes, they start to die. They release IL-1 alpha. IL-1 alpha activates the Kupffer cell that are macrophages that are resident in the liver. It's a very important population in the liver. IL-1 alpha activates Kupffer cell. Kupffer cell, they secrete IL-6, and IL-6 tells to the hepatocytes to start the proliferation. This is key, because if you, in the IL-6 knockout mice, these mice don't develop cancer because they cannot start the compensatory proliferation. So first of all, our question was, is GAD45 beta induced following DNA injection? It turns out that it is. How are the cell death and proliferation affected? We see by tunnel staining, a marker of apoptosis, that we saw reduced tunnel positive cell in the GAD45 beta knockout mice compared to wild type. And this was also confirmed by Western blot by reduced cleaved caspase 3 here. So cell death, it's reduced in the GAD45 beta knockout mice. Next, how is the proliferation? The same, Ki67, a marker of proliferation, less in GAD45 beta, more in the wild type. Cyclin D1, also less in GAD45 beta, more in the wild type. So compensatory proliferation and cell death are both reduced in the GAD45 beta knockout mice. IL-1 alpha and IL-6 are two proteins that connect these two processes. So we went to see in the serum of mice injected with DAN, we notice a trend of decrease of IL-1 alpha, but not significant. However, there is a great reduction in IL-6 in the GAD45 beta knockout mice compared to wild type, consistent with reduced proliferation. We also went to see IL-1 alpha in hepatocytes, and we saw that there was no decrease. But however, GAD45 beta knockout mice show reduced IL-6 in Kupffer cell. So what this suggests is that GAD45 beta somehow can stimulate the proliferation of IL-6 in Kupffer cell. IL-6 bind to the hepatocytes and start the compensatory proliferation. This is absent in GAD45 beta knockout mice. This is actually a pathway for IL-6 because IL-6 activates STAT3 in the hepatocytes. We did a Western blot here and actually saw reduced phosphorylation of STAT3 in the hepatocytes, confirming reducing IL-6 activity. Then we also went to see, okay, how are the immune cell affected in the liver of GAD45 beta knockout mice compared to wild type? We noticed that there are less macrophages by F480 immunostaining, but also less neutrophil by actually flow cytometry. So we say that there is actually less cell death, less compensatory proliferation correlated by a decreased immune infiltration. Next, as I told you before, the main mechanism of action of GAD45 beta is by modulating the MAP kinase cascade. So the next logical step was to check, okay, how is the MAP kinase cascade affected? This was a very good result because we actually had decreased phosphorylation of JUNC that is very important. I'll explain later on. There are two MAP2K that phosphorylate JUNC, MEC7 and MEC4. And we actually found that there was a slight decrease in MEC4 phosphorylation in GAD45 beta knockout mice compared to wild type following DNA injection. So this points to GAD45 beta directly or indirectly regulating JUNC function. And we learned a lot of things by JUNC in the field of hepatocellular carcinoma, because we learned that it doesn't matter the gene, but it's the cell type where you knock out the gene that is very important. If you take actually JUNC knockout mice, you injected them with the AN, you see that they are resistant to tumor. They don't develop any tumor. But however, if you knock out JUNC in hepatocytes, what you see is that these mice develop tumors. But if you knock out JUNC in myeloid cells, and Kupffer cell is the main type of myeloid cell in the liver, what you see is that they are resistant to tumors. So we say that in order for this phenotype to happen, the whole body, the function of JUNC in Kupffer cell, it's extremely important. And we thought that GAD45 beta may actually be a new regulator of JUNC in Kupffer cell. So what we did was that we isolated Kupffer cell from wild type, GAD45 beta knockout mice. We treated them with IL-1 alpha, IL-1 beta that are two known inducers of IL-6 in Kupffer cell. And we saw by ELISA that the level of IL-6 was actually decreasing GAD45 beta knockout mice compared to wild type. And here I show that this decrease is actually associated with a decrease of JUNC in GAD45 beta knockout mice. So that tells us that IL-6 cannot be secreted by Kupffer cell because you lack GAD45 beta. GAD45 beta stimulate IL-6 secretion in Kupffer cell. Then We wanted actually to further prove the model, and would have been nice to have, but we didn't have. It's a model where we lack GAD45 beta in hepatocytes, and we lack GAD45 beta in Kupffer cell, and to compare the model. This was not available. However, since Kupffer cell, they derive from bone marrow, we thought about doing this strategy. We took a wild-type mouse, we transplanted GAD45 beta bone marrow, and therefore we generated a wild-type mouse with a GAD45 beta immune system. Since Kupffer cell are the main type of myeloid cell in the liver, I'm going to refer to this mouse as GAD45 beta delta Cup. We also did the reverse experiment, GAD45 beta knockout liver transplanted with wild-type bone marrow. We generated GAD45 beta delta head, that actually GAD45 beta is lacking in hepatocytes or is lacking in Kupffer cells. So this model enables us to see the role of GAD45 beta either in hepatocytes or in Kupffer cell. We actually injected these mice with the AN, and as we were expecting, only when you lack GAD45 beta in Kupffer cell, but not in hepatocytes, you have a reduced cyclin D1. So you have actually reduced compensatory proliferation, consistent with the role of GAD45 beta in Kupffer cell in stimulating compensatory proliferation. We also checked IL-6 in the blood of these mice, and what we actually saw here is the GAD45 beta, when you lack GAD45 beta in Kupffer cell, you have a reduced production compared to when you lack GAD45 beta in hepatocytes. So this was a very nice result. And so on the basis of what I told you before, what I can say is that, you have the liver, you have two main type of cell, GAD45 beta in hepatocytes and in Kupffer cell. You inject the atyl nitrosamine, you activate GAD45 beta in hepatocytes, you activate JUNC. You actually stimulate cell death. The cell death release IL-1 alpha. IL-1 alpha binds to the receptor on the Kupffer cell. You actually activate GAD45 beta in Kupffer cells. Oh, I'm sorry. You notice that there is this window. I just realized. Then you actually stimulate IL-6 production that binds to the receptor on hepatocytes. We have actually the compensatory proliferation, and the compensatory proliferation leads to HCC development. Okay, so on the basis of this, my conclusion is that we actually identify GAD45 beta as a new regulator of JNK function and IL-6 production in Kupffer cell that in turn affects liver cancer development. So GAD45 beta is required for the induced hepatocellular carcinoma. GAD45 beta promotes hepatocyte cell death and compensatory proliferation in response to the atyl nitrosamine. GAD45 beta control then induce hepatocytes proliferation by upregulating IL-6 expression in Kupffer cell. This is very important. And GAD45 beta promotes JNK phosphorylation in Kupffer cell, eventually leading to IL-6 production. So I have to thank actually my group, that was Jesus, my group leader, Nicolas, he gave me a great deal of help. Vanessa was an exceptional technician. Christian was a great guy, and Maria. And ready for your Q&A, and thanks very much for your attention. Yeah.
I have one doubt actually.
Yeah.
You have shown the mechanism using a done artificial, this one actually. Is the same mechanism follows for hepatitis B, hepatitis C also?
That's a very good question. I don't think it has been studied in hepatitis B. But I cannot say, and this is the key question. As I told you before, we thought that GAD45 beta was a tumor suppressor, given the human evidence, okay? In humans, you cannot inject DAN on a human, right? But you can do it on a mouse.
Mouse model.
So I think the moral of the story that I got is that a function of gene is not only liver cancer, but is the way you induce liver cancer. One thing is if you induce liver cancer with hepatitis B. Another thing is if you induce it with chronic obesity. So the gene can be a tumor promoter or a tumor suppressor in the way you induce cancer. And the only thing I can tell is that in mice with the atyl nitrosamine, that paradoxically, why we use the atyl nitrosamine so much? Because the cancer that they originate genomically and mutationally wise, they resemble the human cancer. However, they resemble, but they are not 100% human cancer, right? And in this case, he is required. But with the other case, it will be good to study. It's a great question.
Okay, thank you very much.
Dr. Humberto, It's not really a question that I have for you. I'd like to congratulate you for your very interesting idea. And I was just thinking aloud that after you uh, apply DN, there is cell death and then cell proliferation.
Yeah.
And then there is the secretion of the Ty2 cytokines, mainly IL-6.
Correct.
And maybe it's forming a vicious cycle of immune suppression, which upregulates the cancer. It's just a statement, maybe, not a question, really.
Oh, I would say I would agree with you. I mean, cancer tends to develop in immune suppression or actually chronic inflammation.
Yes.
That what happens in chronic inflammation is that, as you probably know, the cell get actually exhausted, and this is a key word, exhaustion, because exhaustion equals to immune suppression, because you have the cells there, but they don't work because they are chronically stimulated.
Yes.
And we know that chronic inflammation is extremely bad. Short-term acute inflammation can be good, but chronic inflammation like alcohol consumption, hepatitis C, cirrhosis.
Yes, and TNF-alpha is one of the culprits for chronic inflammation and causing cancer.
Just a last question. I have a last question. Can I ask? Did you look at the methylation pattern of your master protein?
So we actually were really interested in epigenetics or methylation, but in the literature, it was actually reported that at least in human, the promoter is methylated. But I mean, I mean, if GAD45 beta promoter is methylated in mice, I think there may be some data about that. For sure, somebody has done some methylation in the AN. So then it's a data that can be extrapolated from somewhere. Absolutely.
Thank you. Thank you.
Thank you. Thank you so much.
Thank you so much, Dr. Umberto. That was really an excellent.
Thank you.
Thank you so much.
Thank you.
Okay. Our next speaker is Dr. G. Karthikeyan from Savita Dental College and Hospitals, India, and he is going to give a talk on role of innate immune mechanisms in the regulation of inflammatory disease. And I request Dr. Karthikeyan to start his presentation.
Yeah, good evening, sir. I am Professor Karthikeyan from Savita Dental College, and today I'm going to talk about the role of innate immune mechanism, the regulation of inflammatory disease. The inflammatory disease we are concerned is about periodontitis. It's periodontium means the tissues surrounding the tooth. So the inflammation of that periodontal tissue is called as periodontitis. So what's the role of innate immune mechanism in its regulation? We are going to see. So this is a healthy gingiva. The teeth is supported in the alveolar bone. The bone and the tooth root are covered by gingiva. So the tooth is attached to the bone by means of periodontal ligament. So the periodontium is a specialized tissue that both surround and support the teeth, maintaining them in the maxillary and mandibular bones. It includes gingiva, periodontal ligament, two soft tissues, and two hard tissues, cementum and alveolar bone. So periodontitis is now it's recently classified as based on its severity and grading. Generally, it is divided into chronic and aggressive forms, either generalized or localized. So aggressive periodontitis, microbial deposits are inconsistent and it occurs less than 30 years. There will be rapid attachment loss and familial aggregation will be there, but systemically the individuals will be healthy. On the other hand, chronic periodontitis, the microbial deposits are consistent. That is more deposits, more destruction. But on the other hand, aggressive, very minimal deposits, but more severe destruction. The chronic age of occurrence is more than 30 years and moderate rate of attachment loss can be modified by smoking or diabetes, systemic illness, and there will be some local predisposing factors. So here chronic periodontitis, you can see a lot of deposits and inflammation of the tissue with the generalized horizontal bone loss. On the other hand, the aggressive periodontitis, you can see a pink gingiva with very minimal deposits, but patients, all the teeth are mobile and shaky. It's severe bone loss. but deposits are very less. So it's influenced by diabetes, rheumatoid arthritis, or preterm birth, or atherosclerosis. Generally, the periodontal disease or gum disease, the occurrence, epidemiological occurrence is most countries, it's more than 90% have some form of periodontal disease, either gingivitis or periodontitis. In Asia and Oceanic region, if you see in adults more than 35 years old, 67% with shallow pockets, 20 to 30% with deep pockets, and in India, 0 to 7% only has a healthy periodontium. That means nearly 90% has some form of periodontal disease, either gingivitis or periodontitis. So this is the occurrence. And the aggressive periodontitis occurs in 2 to 3% in an old population. So what is the etiology? The main etiology for periodontal disease is gram-negative facultative or obligate anaerobes. Microbes are the main etiology, but race and ethnicity is also an important risk factor for destructive periodontal disease. And more than 500 bacterial species have been identified in the plaque or the tartar or calculus, the deposits, hard mineralized deposits. And among the various bacteria, the gram-negative facultative or obligate anaerobes like Aggregatibacter actinomycetum committens, Porphyromonas gingivalis, Prevotilla intermedia plays an important role in causing the disease. So these are the bacteria. The Aggregatibacter has some internal star-shaped colony. Porphyromonas gingivalis is a black pigmented bacteroid. Tannella, Forsythia, Chikungunya, denticola are all obligate anaerobes. These are the complex community bacteria which causes a severe destruction of the supporting bone. So the risk factors for periodontal disease are smoking, diabetes, stress, genetics, some medication or hormonal changes. And among these, the genetic risk factor plays a major role. And this was found by Kahneman et al. In 1997, a composite genotype called gene polymorphism in interleukin 1 alpha at minus 889 and interleukin 1 beta plus 3954 position. If there is a change in the nucleotide from C to T, those individuals are more prone for periodontal destruction. So a device, a test introduced as PST, periodontitis susceptibility test, for checking for this composite genotype. But when the composite genotype was checked in different population, it was found to be associated with periodontal destruction in Caucasians, but not in Asian region. In India itself, we did study, we didn't find any significant association. So the race and ethnicity is an important risk factor for destructive periodontal disease. So various studies have been done with the cytokines or with the matrix metalloproteinase or enzymes or connective tissue components or signaling molecules, the genetic polymorphisms of between periodontitis group and healthy control groups. And lot of studies have been published, which gives different results. All these studies have seen an individual genes which regulates, which are showing some regulation in the immune mechanism. So we ourselves did a lot of association analysis of individual candidate genes like CTLA4 or TAC1 with vitamin D receptor polymorphism or toll-like receptor polymorphism or interleukin 1 beta or calprotectin, and we have seen so many these things. Some we have found association, some we didn't find association in Indian population. So what is this innate immunity? The host may respond to inflammation to a range of challenges from bacteria to cholesterol. However, the nature of the response differs and its character depends on the microbial triggering of specific receptors, signal transduction pathways, and the way cells and tissues respond to these signals. The challenge to overcome escaping recognition by microbial mutation was overcome by innate system called pattern recognition receptors. So the main pattern recognition receptors are toll-like receptors, which are critical for recognition of microbes by the immune system. The TLRs are expressed on lymphocytes, epithelial cells, osteoclasts, precursors, macrophages and dendritic cells. They recognize pathogen-associated molecular patterns. The PAMPs like lipopolysaccharide, lipoproteins, lipoteichoic acid, flagellin, single-stranded RNA of the microbes. These are screened by these toll-like receptors on various cells throughout the lifetime. 24/7, it's screened. So there are various types of toll-like receptors on various cells. TLR 1, 2, 3, 4, 5, 6, 7. Each has its own function. For example, TLR 2 will always screen for gram-positive organisms, that is lipoteichoic acid, whereas TLR 4 screens for lipopolysaccharide, which is a component of gram-negative bacteria. TLR 9 will screen for CpG motif, or TLR 5 will screen for flagellin, bacterial flagellin. So each component of bacteria or virus or the microbes are screened by these different TLRs on various cells. So the TLR 4 and 2 are more predominantly seen in periodontal tissues, according to Hatakama et al. And this TLR 4, as I told, screens LPS, which is a component of gram-negative bacteria, and most of the periodontal pathogens are gram-negative, facultative, or obligate anaerobes. So this TLR 4 plays a major role in regulating the periodontal destruction. So what are epigenetics? Epigenetics is the study of permutable changes in gene expression that occur without changes in DNA sequence. And a lot of small nucleotide RNAs, like small interfering RNA or microRNA or long non-coding RNAs, plays a role. And inflammasomes are multimeric protein complexes that assemble in the cytosol after sensing pathogen-associated molecular patterns or danger-associated molecular patterns. Activation of this inflammasome is a key function mediated by the innate immune system. So the epigenetic regulation occurs by 3 methods: methylation, or histone modification, or RNA interference. So these microRNAs are playing a major role in the regulation of the inflammatory disease like periodontitis. So where there are more than 2,500 microRNAs, but certain microRNAs are found to play a major role in the regulation of periodontal disease, like microRNA 146A, microRNA 499, microRNA 153, and microRNA 223, or LET-I. So many microRNAs are found. So among this microRNA 146A and 499 plays a major role in the regulation, because this TLR pathway, signaling pathway, once this TLR4 toll-like receptor 4 screens lipopolysaccharide or the gram-negative bacteria, it sends signals through the intracellular component, and through the myeloid differentiation factor, the signal passes. The next component it transfers the signals is tumor necrosis factor 6 IRK, interleukin receptor associated kinase 1. So these will send the signal to the nuclear factor kappa B. The nuclear factor kappa B is the transcription factor which enters the nucleus and stimulates the pro-inflammatory cytokine genes like interleukin 1 alpha or 1 beta, which causes regulation of other cells and post-immune response against the invading pathogens. So this TRAF6 and interleukin receptor associated kinase 1 are major signaling factors in the cell which sends a signal. So what this microRNA 146A is doing, it negatively regulates, it blocks this IRAK1 and TRAF6, whereas the microRNA 499 blocks the nuclear factor kappa B. So both these microRNAs are essential in the regulation of pro-inflammatory cytokine secretion, which in turn is regulating the periodontal disease pathogenesis. So the hypothesis what we formed is major components of innate immune system in recognizing pathogen and activation of further immune process are toll-like receptors and NLRPs, that's inflammasomes, not like receptors. So the genes of these components are regulated by various microRNAs. Thus, we framed a hypothesis that there is no association of 3-UTR untranslated region gene polymorphisms of TLR4, NLRP3, and microRNA 146A with periodontitis in South Indian population. So the aim of this study was to analyze the association of this gene polymorphism with periodontitis. So for this is a cross-sectional study design. We took 328 samples and the subjects were stratified into 3 groups with chronic, aggressive, and controls. And patients were classified based on AAP. The clinical parameters that were accessed to pocket depth, clinical attachment level, block indices, mobility, furcation, and the radiographic assessment. And the DNA was isolated from the venous blood, and the polymorphisms were analyzed using real-time PCR. So these are the real-time PCR graphs of NLRP3 position 10754, 558, and NLRP3 10802501 position, where what changes, or TLR4 position and microRNA 146A position. So the genotype and allele frequencies of NLRP3 polymorphism, we got all the regions. We didn't get any significant association between the control group versus periodontitis or control group versus chronic or aggressive periodontitis. Whereas NLRP3 10754 558 polymorphism with periodontitis, we got a significant, highly significant association as far as the control versus periodontitis was concerned, 0.001 and 0.007 for allele, C allele and G allele, and the G allele is considered a minor allele. And similarly for control versus chronic periodontitis also, we got a significant association, both genotype and allelic. And even with aggressive periodontitis, we got a significant association. So this polymorphism found to play a major role in the regulation of periodontal disease pathogenesis. And similarly, the genotype and allele frequency of TLR4 didn't show any significant association. Whereas the microRNA 146A per 2910164 position, the G and C allele showed a significant association, with C allele playing a minor allele role and G allele as a major allele, and found to have a significant association with chronic periodontitis. So based on this, we are seeing that the microRNA 146A polymorphism and the inflammasome NLRP3 polymorphism is having a major role, whereas the toll-like receptor, the receptor on the cell surface, both the polymorphisms didn't show any significant association. Thus, the intracellular component, the inflammasomes like nod-like receptor polymorphism, and the epigenetic regulators like microRNA, which plays a major role, the genetic changes showed a significant association with deceased individuals compared to healthy controls. So thus, the NLRP3 G allele and the microRNA 146A G allele showed to be significantly associated with the inflammatory disease like periodontitis in Indian population. So thus, I would like to conclude that the 3' untranslated region gene polymorphism of nod-like receptor NLRP3 inflammasome and microRNA 146A do confer risk for periodontitis in South Indian population. Thank you.
Yes. Thank you, Dr. Karthikeyan. Thank you for the excellent presentation.
Thank you, sir. Thank you. Yeah.
The inflammasomes, the polymorphism we observed is at the 3' UTR region, not at the promoter or the coding sequences, etc.
Not in the coding sequence, sir. It's in because these microRNAs are going and binding at the 3' UTR region. So this microRNA has role in both NLRP3 and TLR4. So we have chosen a microRNA like that, and we have seen a microRNA gene. It is in the seed region. That seed region, what polymorphism is there, whether any association. TLR4 and NLRP3 3' UTR region.
They form loop-like structures. Am I correct?
Yeah. Yes.
Loop-like structures.
Yeah. Yes, sir.
Okay. Compliments for the presentation. Was great. And mine isn't really a question. It's more about the prognosis for periodontitis, because if you have a mild grade periodontitis where your teeth start shaking, I mean, what happens? Is there a way to stop them shaking, or they have to be extracted?
Yeah. There are a lot of recent surgeries, are there regenerative surgeries. So we can save only very extremely mobile and very shaky teeth. In a terminal stage only we are removing the tooth. Otherwise, we have a lot of surgical techniques and advancements in the regenerative materials are available. So we can save tooth. Very extreme situation only we are removing tooth. Otherwise, we are shaving all the tooth. That's my specialty.
And one last question about this is more a perspective. It's you know, in dentistry, you know, the dream of everybody will be to regrow the tooth rather than to extract it or to cure some. There will be the holy grail is to tooth regrow.
That side it's going, stem cell research or regenerating pulp through this thing. That's also going, but not in very this thing. It's in a research stage. It's not clinical usage stage.
Yeah, okay, but we are getting there, right?
Yeah.
Great.
Yeah.
Thank you.
Okay. Thank you so much, Professor Karthikeyan.
Thank you, sir.
Thank you so much. Yes. Moving on, our next speaker is Dr. Yakub Matai from Marma Health Center, India. He is going to give a talk on the purpose of temperature of fever in COVID-19. And I request Dr. Yakub Matai to start his presentation.
Okay, I am ready. I want to share my screen. Okay.
Yes, please continue.
Yes. The purpose of temperature of fever in COVID-19. I am Yakub Matai from Kerala, India.
Cold temperature వస్తుంది కదా. Sorry.
All human beings and animals and amphibians affected by fever plenty of times in their lifetime. Even though now we are living with a lot of disease related fever with different names like Chikungunya fever, West Nile fever, rheumatic fever, etc. One question is, are there different types of fevers or is it just a fever? There is only one fever. Definition, diagnosis, and treatment of fever is the same. The symptoms, signs, signals, and actions of disease is different, but the symptoms, signs, signals, and actions of fever are the same. That means there is a scientific basis which is common for all these diseases related with the fever. We can use of all diseases with the fever. Diagnostication of separating fever and disease. Let us examine the present state of fever. One medical book says, our understanding of the neural basis of thermoregulation and fever is still rudimentary. The role of fever in defense reaction is not clear. In practice, as with the pain, relief from fever with the drugs adds to comfort of the patient. It also impresses the patient and the relatives favorably about the therapeutic capability of the doctor. First question is, how can fever be treated with rudimentary knowledge? The fact that the patient does not know that the fever is being treated by rudimentary knowledge is the basis for the survival of the physician. A scientific approach is necessary to evaluate and treat fever. My research is to find out the purpose of rise of temperature in the case of fever. different type of fever definitions. Different types of fever definitions can be seen in various medical books. The following 8 questions need to be answered to know about the fever. What are the ingredients necessary to develop a fever and hyperthermia? Second question is, what are the ingredients necessary to curing a fever and hyperthermia? Third question is, what is the purpose of temperature of fever? Fourth question is, why is the temperature of the fever not above 42 degrees Celsius? The fifth question is, the heat energy used for which activities in fever and hyperthermia? Sixth question is, what are the problems not knowing the purpose of fever? Seventh question is, what are the difference between actual fever and the temperature of fever? Eighth question is, during fever, why our body acts against the facts of physics? No medical books give clear answer to above 8 questions. Necessary ingredients to develop a fever and hyperthermia. Modern science says that fever is an inflammatory response that extends beyond the site of infection. The right ratio ingredients like inflammation and pyrogen is necessary to develop a fever as a good health. Inflammation and pyrogen never cause hyperthermia. All that is needed for hyperthermia is uncontrolled heat only. Many people who have infections in the body do not develop a fever. Many people who have diseased organs may not develop a fever. This is lack of right ratio of development of fever. Necessary ingredients to cure fever and hyperthermia. For curing fever, we should know what are the ingredients necessary to develop a fever. Despite the correct ratio ingredients of fever will decrease the intensity of fever. By removing the correct ratio ingredients of fever will cure the fever. If the inflammation is reduced, the fever will go away. The only way to change hypothermia is to water and reduce the heat. Hypothermia does not go away because the inflammation is reduced like as in fever. What is the purpose of temperature of fever in COVID-19? When the disease made by SARS-CoV-2 virus 2 becomes a threat to life or organs, blood circulation decreases, temperature of fever will emerge to increase, providing essential blood circulation, and it acts as a protective covering of the body to sustain life. It is an adaptation. In all diseases which decrease essential blood circulation and temperature, fever will emerge to increase essential blood circulation and temperature. There is no way other than this to increase, providing essential blood circulation for a sensible and a discrete immune system to protect the life or organ. Modern science says fever is an immune response against infection. If fever is immune response against infection, temperature of fever is not a surplus temperature. It is not supposed to be eliminated from the body. If you try to eliminate the immune response against infection, the infection will increase. It is dangerous to immune system. It is necessary to protect the immune system to increase essential blood circulation. Never allow body temperature to lose by atmosphere. A Brody hen does not, a Brody hen's body temperature increase like that during fever to increase essential blood circulation in the body. The Brody hen does not eat any food. Brody hen's food is a heat which helps to convert fat into energy. Many people who have misconception that fever should be cured. It is not the fever that need to be cured. It is the disease or cause of disease that need to be cured. Fever is an adaptation only. Fever can be compared to our soldiers. Sometimes our patriot soldiers may be defeated, surrender, or even lie. It is not due to the anti-national activity of our soldiers. New fever definition. If essential blood circulation increases to organs, fever is a sensible and discrete action of immune system to increase essential blood circulation as a self-defense mechanism of the body to sustain the life. We can answer almost all the questions about a fever with this definition. Why is the temperature of the fever not above 42 degrees Celsius? To know the answer to this question, you need to know what the temperature of fever is for. When our body is over 40 degrees Celsius, our immune system knows that vitamins, proteins, and other elements will lose their proper form and perish. And need up to 42 degrees Celsius to save the life and organ until the last minute. So our immune system creates temperatures below 42 degrees Celsius. When the hen is brooding, the temperature of hen does not exceed the limit because the hen's immune system produces heat with a clear purpose. Once that purpose is accomplished, that increased heat will disappear or the production of that extra heat will stop. Similarly, our immune system created temperature below 42 degrees with the clear intention of saving life or organ until the last moment. Once that is accomplished, our immune system will stop overheating. The temperature of fever increase in proportion to the increase in inflammation in our body and the decrease in blood circulation. Inflammation is a condition in which the temperature of fever increases as blood circulation decreases. Similarly, as the inflammation decreases and the blood circulation increases, the temperature of the fever decreases. The heat energy used for which activities in fever and hypothermia? Increased blood circulation to most important organs. In disease increased fever, the activity of most important organs increases and the activity of less important organs decreases. This is not due to the increase in temperature, it is due to the decrease in essential blood circulation. This is like an inverter used to work most essential lights and machines in an emergency due to power failure. During fever, our body increases activities of most essential organs like brain, heart, kidney, and liver. Uncontrolled temperature can occur in heat stroke. It is not below 42 degrees. Moreover, it was not created for a specific purpose. When our immune system does not fix like a water sponging that reduces the temperature of fever, the body resists and hates it. The body never likes the temperature of heat stroke. And the body likes all the activities like sponging with the water that reduce the temperature. When there is a fever, the body like and accept the temperature up to 42 degrees from outside and inside to the body. Similarly, the truth can be identified by examining the body's aversion to temperature above no fever. Why do fever have symptoms, signs, signals, and actions? Fever is an action that has symptoms, signs, signals, and actions. Keeping the temperature below 42 degrees Celsius is just an act of fever. Another action of fever is the action of TNF-alpha, a chemical that causes bitter taste and decreased appetite. Why does not everyone with the disease have a fever? 3 reasons. First reason is virus, bacteria, fungi does not cause fever in everyone because the cause of disease does not decrease essential blood circulation to the organs. Second point is the body does not need fever protection. Third point is the immune system does not have energy to produce the temperature of fever as in AIDS. How can create a fever and hyperpyresia and fits within hours? In people with tonsillitis, children and the elderly, using ice water, we can create a fever and hyperpyresia within a few hours. With the continued use, we can cause death in them. Talikutal is the best example. Talikutal is the traditional practice of senicide, killing of the elderly, or involuntary euthanasia by their own family members, observed in some parts of southern districts of Tamil Nadu, state of India. This is a process in which an extensive bath is given to an elderly person before the crack of dawn. He or she is given several glasses of cold tender coconut water and head massage with cold water. By doing this, the temperature of the body falls suddenly, causing high fever, which leads to the death of old age person within 1 to 2 days. Similarly, in COVID patients, using sponging with ice water, we can create a fever and hyperpyresia within few hours. With the continued use, we can use death in them. Prolonged exposure to water can cause fever. How unscientific it is? How unscientific is it to say that you can change that fever by dipping water in your body? Some thought-provoking questions and answers. If pouring cold water on the body can reduce body heat and cause fever, hyperparesia in Talekutal, Cannot any disease cause fever and hyperpyresia by pouring water on the body? The answer is can. Second question is there any difference between a fever caused by Talaikuttal and other illness? The answer is no. Third question is there any difference between fits and death due to heat loss in Talaikuttal and fever, fits and death due to heat loss in COVID-19? The answer is no. Is there any difference between COVID-19 fever treatment and Talaikuttal fever treatment and any other fever treatment? The answer is no. Is conservative fever treatment suitable to Talaikuttal? Answer is no. In Talaikuttal, what are the necessary ingredients to cure hyperpyresia? Answer: to increase blood circulation and to decrease inflammation, heat supplementation is necessary and heat loss from the body. If fever with Talaikuttal can be cured with the heat, can fever with any diseases not to be cured with the heat? The answer is can. Next one, if water in the body can reduce body temperature and cause fever, then water again can reduce temperature of the body and cause fits and death. As in Talaikuttal, if modern fever treatment can be done by sponging or immersing the body in ice water in hyperpyresia, can it not cause fits and death by reducing the body temperature? The answer is can. Most medical books says patients with the hyperpyresia should be immediately immersed in ice water regardless of the cause or disease. According to this, in patients with hyperpyresia due to Talaikuttal, does immerse in ice water cause death? The answer is there will be death. Next question is in the signs of fever, fits and death in Talaikuttal the same as the signs of fever, fits and death in other diseases? The answer is yes. Will assisting fever treatment reduce COVID death? The answer is no. Did any patient die without developing a fever COVID-19 and without a heat reduction treatment for that fever? Answer is unlikely to occur. How unscientific it is said that prolonged exposure to water can cause fever and that fever can be cured by sponging it in water. Fever is measured with a thermometer and hyperthermia is measured with the same thermometer. This is purely unscientific. The basic element necessary for a scientific treatment is not provided in fever treatment. How can we prove that the temperature of fever is to increase essential blood circulation? If we ask any type of question related to fever by assuming that the temperature of fever is to increase essential blood circulation, we will get a clear answer. If we evade from this definition, we will never get a proper answer to even a single question. If we do any type of treatment by assuming that the temperature of fever is to increase essential blood circulation, the body will accept at the same time, body will resist whatever treatment to decrease blood circulation. If we measure the heat energy used for which activities in fever, we will know the purpose of the temperature of fever. It is astonishing that modern science has not been able to examine what actions with the heat energy of fever despite its advanced development. No further evidence is required to prove the temperature of fever is to increase essential blood circulation. Apart from this, what kind of evidence is needed to prove that the temperature of fever can increase blood circulation? That is a question. Let me know no parameters to prove the purpose of temperature of fever. We can prove that the temperature of fever is to increase essential blood circulation with that parameters. Fever is a protector or destroyer. Will temperature of fever protect the brain cells by increasing essential blood circulation to the brain, or will it destroy the brain? When disease increases, essential blood circulation energy level also decreases. The vertical height between heart and brain is more than 1 feet. When the disease becomes severe, ability to pump to the brain decreases. Then blood flow to the brain decreases and delirious of fits are formed. As a result of this, brain cells are damaged, so the patient might be paralyzed or even may die. Fever and delirium. Fainting or delirious or damage to the brain cells is not due to the increased temperature of fever. It is due to decrease in blood circulation to the brain. Without knowing the purpose of fever and without examining what actions were taken with the heat of fever, it is said that fits or delirious would happen to increase in temperature of fever. Temperature below 42 degrees never cause any harm to our body. During summer in some parts of India, atmospheric temperature is more than 45 degrees centigrade. Besides that, a sauna temperature ranges from 160 to 200 degree Fahrenheit. In a steam bath, a person being 15 to 25 minutes. While taking a normal steam bath, a person inside the bath is more than 50 degrees centigrade. There is no such a history one had faded or affected with the delirious or fits. During fever, if you take a steam bath, fits never happens. Blood circulation never decreases. Skin never shrinks as in fever. Signs of curing fits, regaining consciousness even before the temperature subsides. When a fainted patient lies on the floor, the vertical height between heart and brain decreases. So blood circulation increases to the brain. And so fits is cured even before temperature subsides. Many people misunderstand that regained consciousness is due to the reduction of temperature. Shivering is to increase blood circulation. Hyperbarometric temperature is below 42 degrees centigrade. Only 4 degree increase in body temperature. If a 4 degree increase in temperature occurs, there is no history to show that shiver occurs. Animal behavior, advantage of instinct. Buffaloes does not try to enter or lie in water, nor drink it. Buffaloes manage body temperature by immersing their body in water and drink it. But when they are having fever, they do not try to enter the water nor drink it. Ways to eliminate fever by increasing blood circulation. Just listen to the messages from the body and act accordingly. Increasing essential blood circulation is the right treatment for fever. Blood circulation never increases without increasing temperature. Fits can never be cured without increasing essential blood circulation. To increase essential blood circulation, never allow body temperature to lose by atmosphere and apply extra heat from outside and inside to the body. It will help to immune system to fight against infection. Blanket, steam bath, hot sandbag or thermal heat pad, hot pepper water, hot drinks can be used. Mechanism application of heat. When the temperature produced by the body due to fever and heat which we applied on the body combines together, the essential blood circulation increases. Then immune system will stop to produce heat to increase essential blood circulation, and the body will get extra heat from outside without any usage of its energy. Heat is the fast and efficient remedy to reduce inflammation and increase blood circulation. During fever, 99% of disease or patient shows inflammation. Apply heat from outside and inside to the body. Blood circulation, inflammation related to fever and its signals, symptoms, signs, and actions will decrease. Then essential blood circulation increase, inflammation decrease, digestion increase, body pain diminishes. Like and upset the hotness. When we apply heat from outside and inside to the body, all signals and symptoms of fever like and upset the hotness according to the directions and commands of immune system. If fever is immune response against infection, our body like and upset the hotness. At the same time, our body dislike and hate temperature reducing methods. If fever is immune response against infection, temperature of fever is not a surplus temperature. If you reduce temperature by water sponging, infection will increase and it may be dangerous. Fever is not a part of disease, it is a part of self-defense mechanism of the body and it is an adaptation. A disease does not require a fever, but at the same time, a fever requires a disease. Both the disease and fever are different. We cannot include body destructing disease and body protracting fever in the same group. Conservative fever treatments and its effects. Fever treatments, role of spongy and paracetamol. Whatever happens to a brooding hen and its egg is sponged with water. The same happens to a fever patient if temperature is decreased by sponging. The same thing happens in Talaikuttal. The researchers found that the glutathione level were lower when single dose of paracetamol was used. This may increase inflammation level. Removal temperature of fever by sponging or paracetamol is not helpful for curing disease. It is believed that the doctor will save the patient in the treatment of fever, but the doctor becomes the executioner who kills the person. All conservative treatments for fever not only do not make any radical cure for the feverish condition, but also further decrease the essential blood circulation and increase inflammation and leads the body to more dangers. The best way to reduce COVID death rate is ban conservative fever treatment in Talaikuttal model. If temperature is decreased by sponging or paracetamol, disease or cause of disease or cause of fever is never decreased. It is proved that fever medicines are more dangerous than disease, and fever treatments are more dangerous than medicines for fever. A lot of problems emerge not knowing the purpose or temperature of fever. It may cause death. Various hypotheses, contrary fever treatments, etc. The result of not knowing the purpose or temperature of fever. Contrary fever treatments, various types of sponging. Some physicians recommend sponging with hot water. Some physicians recommend sponging with normal water. Some physicians recommend sponging with ice water. Most medical books say that patients with hypertension should be immediately immersed in ice water, regardless of the cause of disease. If the patient is shivering, all the physicians recommend hot water sponging. Most patients are troubled when they have hyperparesthesia. If temperature of the surplus temperature or it is supposed to be eliminated from the body, the ice water is must to reduce body temperature. If fever is immune response against infection, never use temperature reducing methods like sponging. Use of ice water is a modern version of Talekutal. If fever is body's own disease fighting mechanism, how does various types of water help into doing it? That's the question. Other contradictory definitions can be seen in various medical books. One medical book says fever destroys homeostasis, body's own disease fighting arsenal, beneficial effects of fever, the adverse effects of fever, fever increase immunity for, fever decrease immunity for, all are seen in various medical books. If the fever destroys homeostasis, fever is always dangerous, so it is eliminated from the body. If the fever is immune response against infection or body is on disease fighting arsenal or fever increase immunity power, it is always good to the body. Some instructed to use air conditioner or fan. Some physicians prohibited to use air conditioner or fan. Everyone in the ICU uses air conditioner. If temperature of fever is a surplus temperature, or it is supposed to be eliminated from the body, air conditioner is must to reduce body temperature. If the fever is immune response against infection, never use fan or air conditioner. Never allow body temperature to lose by atmosphere. Another contradictory treatment is blankets. Some physicians are restricted to use blankets. Some physicians are prohibited from using blankets. If the patient is shivering, all physicians will the patient to use a blanket. If temperature of fever is a surplus temperature, never use blankets. Blankets increase the body temperature. If fever is immune response against infection, always use blankets. Another contrary fever is drinking water. Some physicians instructed drink water. Some physicians are recommended drink cold water. If patient is shivering, all physicians are recommended drink cold water. Most patients tremble when they have hyperparesthesia. If temperature of fever is surplus temperature, always use cold water or ice water. Never use hot water. It will help to increase the body temperature. If fever is immune response infection, always use hot water. After analyzing and evaluating the statements and correlate them, it will be clear that what is right and what is wrong. Besides that, it was found that all statements in the modern medical books were contradictory, leading us to utter confusion. As science cannot have such a conflict in practices. If we read the contradictory definitions and treatments in front of a rock statue, it will start to laugh. Is there any base to conservative fever treatment? All treatments of fever are based on belief that the fever temperature between 38 to 41 degrees harm to brain and body. At the same time, there is no concrete evidence to this belief. The purpose of rise of temperature is not known. Without knowing the purpose of the temperature of fever and without examining what action was taken with the heat energy of fever, what science can be used to say that the fever is dangerous? Without aim and target for rise of temperature in the case of fever has been subject to immunology research was by a lot of medical universities in medical international repute. But unfortunately, the modern medical science have concluded that the purpose of temperature could not be found. Importance of finding the purpose of temperature of fever COVID-19. Immediate relief from fever and body pain. Life-saving discovery. A single magic answer to every fever-related questions. If medicines are prepared according to the purpose of temperature of fever, any country can guide the world in the cure for fever. Conclusion. When disease becomes threat to life or organs, the temperature of fever in COVID-19 is to increase essential blood circulation. Fits, deliveries, or damage of brain cells are not caused when an increase in temperature of fever takes place. It is due to decrease in blood circulation to the brain. Apply extra heat from outside and inside to the body will increase blood circulation. Conservative fever treatment should be discrete action immune system. There is only one way to reduce COVID death. Prohibit Thalikothal model conservative fever treatment. There is no other way than this. It is powerful that a physician having years of experience in fever treatment cannot understand actual purpose of fever. I have developed 8,000 affirmative cross-checking questions. It is proves that fever is to increase essential blood circulation. In the case of fever, double-blind study and placebo cannot be done properly because the patient will quickly recognize this when experimenting with the heat instead of using cold as a way to reduce the temperature of fever. Please note that 9, 10, and 11. This is belong to the Thalikothal. Thank you. Hearing me patiently so far. If you have any questions, you can contact my personal email ID and my WhatsApp number. Hello.
I have a question for Dr. Mathai. Dr. Mathai?
Yes. Yes.
It's just an ethical question that since euthanasia is not allowed in India, so is not Thalikothal punishable by law in India?
Yes. Punishable. Now it is Thalikothal now punishable in India. Already parliament now it is done various parts of Tamil Nadu. In 2019, there is a
You're not audible.
It is punishable in India. Parliament is passed this law, but in some parts of Tamil Nadu, it is done. Now it is also a custom. In 2019, there is a cinema produced in Kerala. Its name is Fever. It is belonged to the Thalikothal.
But if it is reported, it should be punishable.
It got a FIPRASI. Yes, it is got a FIPRASI award in last year for that cinema. It is punishment,
No,
but it is also done there.
I'm not talking of the cinema award, but I'm just asking that if it is reported, it should be reported if it is detected, and it should be punishable by law. It's just an ethical statement.
So many medical studies are done about this. So many medical studies. Please check the reference number 9, 10, and 11. All are belonged to the Thalikothal. We can create immediately create a fever, hyperparesis, and death within hours.
Thank you.
Thank you so much. Yes.